So you're live now, please start.
Yeah. Good afternoon, everyone. Good afternoon to all. Once again, welcome back to NPTEL online certification course on novel drug delivery systems. I, Dr. Sathish Dyawanapelly, working as assistant professor at Department of Pharmaceutical Science and Technology, Institute of Chemical Technology, Mumbai. So here, my TA is also available here. So he's introduced himself, and then we'll start the live interaction session. Yeah, Rushikesh. So Rushikesh is working under me at ICT Mumbai. So he's working on the various novel drug delivery systems. So he's developing various types of nano formulation, including polymer
Hello,
micelles. Also, he's working on liposomes and the development of various industry projects also. Yeah, Rushikesh.
hello, I'm audible, sir.
Yeah, yeah.
Hello, everyone. I'm Rishikesh Mahendra Singh Girase, TA for Novel Drug Delivery Systems. Currently, I am working as a project assistant at ICT Mumbai under Dr. Sathish. The work is depends on the novel drug delivery systems. So, I am developing liposomes, neosomes, then microemulsions, and nanosuspensions, and also working on various conventional drug delivery systems. So, that's all about myself.
Yeah. Thank you, Rushikesh. So, yeah, so he's helping in various content development, also in the editing of the videos and also preparing of the assignment. And not only that, he's helping of the compiling of every data which we are working for this NPTEL course. And also we acknowledge even IIT Bombay team, so they are continuously supporting us for the editing and also the recording the videos. And we welcome once again to all. So we can discuss if you have any some questions in the live chat. So those who are interested, you can put in the questions. We will discuss. So we have couple of questions. So which students are requested. So first, yeah, first question, one faculty, we requested Anamika Daniel, this faculty. So suggest the preparation of the nanoparticles. Yes, these have extensively we have discussed in the unit week 2 and week 3. Yes, first of all, we need to understand that. So we have various types of the nanoparticles, right? So which can be classified into different categories. That is into the polymeric nanosystems, also the lipid-based nanosystems, and third one is the inorganic nanoformulations. So definitely this preparation method will vary from one formulation to other formulations. When you select in the polymer, so we have again in the polymer, we have different types of polymers. There are the polymers which are Yeah, I'm sorry, some technical issue. So I hope, can you hear me? Yes, Rishikesh? Yes, okay, thank you. Yeah. So yeah, so we are discussing on the preparation of nanoparticles. So it depends on the type of polymer. If it is a hydrophobic, we need to select solvent-based methods. If it is a positive charge, we need to look for the negative charged opponents. If it is a negative polymer, we need to select positive charge. So it depends on the properties of the polymers. So again, in the polymer nanoparticle, we have various techniques. So in that we have ionic gelation method, solvent evaporation method, solvent emulsification method, and also we have the diffusion method, we have double emulsion method, right? So we have various methods. Again, it will also depends on the type of drug molecule. If a drug is water soluble, we need to select different method. And if it is hydrophobic, we need to go for some solvent emulsification or solvent evaporations methods. Okay. So there are the scalable methods. When you are going for industrial scale, you need to select the scalable methods like using of the supercritical fluid technology or by using of high pressure homogenizer. So we need to use scalable tech. When you coming to the lipids, okay. So in the lipid, again, we have different lipids, right? We have different category of the lipids, right? We have liposomes, we have lipid nanoparticles, right? We have NLCs, we have solid lipid nanoparticle, and again we have different techniques. Okay, again it depends on the type of uh, carrier and as well as type of drug. There are the drugs which are comes in the like water soluble. Such cases we have to select the right method, and there are the cases when you use water soluble drugs, the encapsulation is the one of the major issue, right? Such cases we need to identify what is the best method by comparing of various techniques, so we can develop the carriers. Now coming to the inorganic, again, we have a iron oxide, we have a silica nanoparticles, right? We have gold nanoparticles, right? So again, most of the cases we prefer precipitation method. When you go for the silver nanoparticle, it comes with the reduction method, that we have various types of techniques. But end of the day, and what is your application, that is also important, right? If you are going for biomedical application and for the drug delivery application, you try to avoid, especially in case of inorganic, you need to avoid the usage of the solvents as well as the as well as the additives or whatever the ingredients that should not cause the toxicity of your materials. So this is about the first question, that is a preparation of nanoparticles. So we have elaborately discussed all types of the methods in during the sessions. You can refer that presentations, as well as we have given the reference in each presentation. You can go through that review article. You, if you come up with one any problem which you are facing with the understanding, that we can discuss in our upcoming live sessions. I hope I have answered your questions. Yeah, so first we'll check if any live questions, then we'll look into that. Yeah. So yes, people are responding. So kindly, yeah, okay. Week 3 assignment question number 3. Rushikesh, can you help me? What is that question number 3?
Yes, sir. It was
Okay.
about the types of
Uh-huh.
analysis. So we have mailed the particular person about the issue, and
Okay. Mm-hmm.
in
Yeah.
accordance with Omkar sir's suggestion, we have taken the action, sir.
Okay. So what is that question 3? Just could you please explain me? If they want, once again, we can help them.
Okay, sir.
So anyone, if you have any doubts, you can ask in the
So
live chat. Yeah.
the question was, which of the following is correct statement for different types of analysis?
Okay.
And we have given four options. First was type 1 NLC, which
Mm-hmm.
is amorphous. The option was
Okay.
amorphous. Then Type 2 NLC is multiple type. Type 3 was NLC imperfect, and type 4
Mm-hmm.
NLC was crystalline. So there is no
Okay.
type 4 election, type 4
Mhm.
NLCs.
Yes,
So
yes.
all the
Yeah.
four options were wrong.
Okay. Okay. So
Yes.
I think we responded in the through mail also, right?
Yes, sir. Yes, sir. Yes, sir. We have.
Yeah. So yeah. Okay, great. So any other questions? Can you see in the live chat, Rushikesh? Anyone from any
Give me a second.
person?
No, sir. I cannot see any doubts.
Yeah. Okay. So then we'll go for another question from the uh, Google form. So yeah, participant, I will suggest, I request you to drop your text or whatever question you are facing or difficulties in understanding. You can put it in the chat box. We will consider your questions. Okay. So yeah, so second question is that explain as well as in Hindi also. Actually, I am sorry for that because I teach in Hindi. There is no issue at all. But in regular classes, in the live classes, I do. But when it comes from like a professional language, okay, so it's very difficult to because we need to consider the all the regions, but that will interrupt with the other language barrier. Yeah, so I can speak in Hindi, but this is the barrier which we cannot do. But if you have any difficulties in any understanding, we will definitely help you. You can contact me through mail or any even in the live session. We definitely will try to help you in understanding in Hindi language also. Yeah, so yeah, that is a requirement. We cannot speak in Hindi. So yes, but you will get transcripts in Hindi for better understanding. By using of the transcript, you can translate into Hindi. You can learn. We are sorry for that. Yeah. So even NPTL will not allow for that. Yeah. So yes, Abhijit Singh is asking question about please add some animation video if possible for better understanding of the topic. Okay, yeah. So I agree with you, but yeah. So right now we have not prepared any videos, but we are planning extension of this workshop. We are planning to make a workshop on the novel drug delivery system if time permits, and if everything goes well, we'll come up with the workshop. There you will get the details of each experiment. We are planning for all type of novel drug delivery system. Yeah, so because of the continuous preparation of the slides and the lectures, so that's why we could not get time for the animation videos. But definitely, so there is a I will put it in the forum. There is a website. There you will find all type of scientific, especially in the pharma, so which you will see the animation videos as well as the practical experiments which are comes in the videos. I think that is Joy TV. I think, right, if I'm correct, Rishikesh. So
Yes,
there
sir.
are
Yes.
the
We can check
people
in.
who uh, so what is that? There is a link, right? Joy TV or something. So they will give all type of scientific videos, recorded videos. You can prefer that scientific videos. I could not get it, but one I can we can drop Rishikesh, note down this comment. We'll send them a link so they can also connect into the videos.
Oh, sir. We can share.
Yeah. So any question from YouTube, Rishikesh?
Yes, sir. I can see one question.
Yeah, yeah.
I will
Please tell me.
read it out for you. How do we evaluate safety concerns like neurotoxicity in brain-targeted drug delivery systems?
Yeah. Come again. Question.
How do we evaluate safety concerns like neurotoxicity in brain-targeted drug delivery systems?
Safety concerns of
In brain
neurotoxicity
targeted.
in brain target. Okay, it's a very good answer. So Yeah, so yeah, first of all, you need to, yes, definitely, whenever you are developing any formulation for not in brain, any type of formulation or any new development, okay? So when you're using any new polymer or any new drug or combination of these things, okay? So you have to prove the safety study, nothing but. So ideally, what we do in a simple, for example, if I'm making any formulation which is I want to target for the brain, okay. So consider I am making a polymer nanoparticles, okay. So I have loaded some the anti-Alzheimer drug. Just I want to give through intranasal route, okay, right. So such cases, what I do, I prepare a formulation. I will check my cell viability by using of the MTT assay. There I need to prove that my only formulation without drug, okay, it should not kill any type of the cells. That is called a viability. So that means your percentage of cells should be viable more than 80%. If comes 80% below, that means your formulation, or you can say your carrier, is the toxic to the cell. So how we do this, okay, based on your routes of administration, okay, right? You need to select the cells. For example, if I'm going through oral route, I will take the any the GIT cells, okay. If I'm going for the ocular, I will take some retinal cells, okay, right? And also, if I'm going for the nasal, I will take the nasal cells, okay, human. You will get all this in the from various cell culture labs. You need to procure the cells from the human cells, okay. If you're targeting for retina, you can take the human retinal cells. And if you're targeting for the human colon, then you can take colon cells, right? So similarly, so if I am targeting for nasal, I will take nasal cells, human nasal cells, and I will see the toxicity with the only my formulation without any drug. In other way, in second step, what you can do, you can load your drug, and again you can see the toxicity. And remember this, if you are targeting for anti-cancer therapy, there you need to prove the cytotoxicity, right? But your formulation, blank formulation without drug, it should not show any toxicity. That is the first condition we do the safety studies. Okay. Now coming to your question, safety concerns on the neurotoxicity. Now you can take neural cells. For example, you are working on the glioblastoma. Okay, right? So we use we use paclitaxel, okay. We use the various types of anti-cancer, it may be docetaxel, other drugs which showing the cytotoxicity on the glioblastoma cells, okay, right. So what you can do, you can take gliomal cells, okay, you can do the toxicity on that gliomal cells. If you want to see the neurotoxicity, definitely you can take neuronal cells, and you can compare your toxicity profile or safety profile of your blank formulation without any drug, and then again compare with your drug loaded formulation. Then there you will see the difference. If your drug or if your formulation, blank formulation, drug loaded, is not showing any toxicity or nothing, but it is not killing the cells below the 80%, that means your product is safe, right? So the safety study is nothing but cell bioavailability studies. And again, there are the we do histopathological studies also. After routes of administration, for example, I am passing my formulation to intranasal route. What I do, I will take the after administration of the formulation to the animal. Then I will take that nasal mucosa. Then I will study histopathology of my nasal cells. If any damage is happening, nothing but my cells undergoing the toxicity or some destruction. So I need to avoid such damage of the cells. Okay. So similarly, whenever you want to do the neurotoxicity study, you can take the cell lines, or you can take the tissues or cell cultures, and you can treat it, and then you can see by using microscopic images. Also, you can do the histopathology, and also you can see the cell bioavailability. Okay. Yeah, so this is the way we do the safety studies. And if you go further higher level, and if you want to see the biocompatibility of your formulation, you can treat with RBC, and you can see the percentage of hemoglobin release. That is nothing but the hemolysis study. Okay, right? You can see the protein aggregation studies also, right? So these all comes into the safety parameter. But as for pharmaceutical application, so most, if you can see many research papers, people do the cell viability studies by using MTT. That will give the safety of your formulation on the cells. I hope I have answered your questions. If you have more doubts, you can note down and you can ping on the chat box. Yeah, Rushikesh.
Yes, sir. The another question is,
Yeah.
how much dosage of green synthesized chitosan nanoparticles should I add in add if I make 200 ml of syrup?
1 second, I will see the question.
Yes.
Who asked this? I could not see in the.
The question is, someone have synthesized green synthesized chitosan nanoparticles and they want to make a
Uh-huh.
syrup out of it. They they want to know how much of green synthesized chitosan nanoparticle they should take to prepare 200 ml of syrup.
I could not see the actually uh, chart box actually. 1 second. Yeah, now I can see. Uh, how much dose of green synthesized chitosan nanoparticle should I add if I make 200 ml syrup? So, yeah, so no, you need to understand the percentage of active ingredient. That is very, very important, because, for example, you are making a, try to understand in this way. If you are making chitosan nanoparticles, now you want to add in the syrup of 200 ml. Okay, so syrup is completely different. Don't confuse with your understanding. Okay, so ultimately, how much you want to provide to the patient, that is important. For example, if you have a formulation, okay, consider you have a chitosan nanoparticle powder, okay, and you want to give 5 mg per ml, or you can give 5 mg of your drug loaded chitosan nanoparticle to the patient in a 5 ml. Okay, right? So you have to calculate your total dose which you are going to distribute in a 200 ml. Okay, right? Consider you want to give 5 mg per 5 ml. Okay, so calculate accordingly. Okay, right? So for 5 ml you have to give 5 mg, right? So you need to disperse your chitosan drug loaded green synthesized nanoparticles according to the requirement. Okay, so for example, if you want to go for 10 ml, so it will become 10 mg. I hope you have clear. And then you can, it will comes in 10 ml, right? And you have to give 20 mg for 1 second, 100 mg for 100 ml, right? You have to give 200 mg in a 200, no, excuse me, 1 second. So consider you are considering 5 mg, okay, per 5 ml, right? So, so you have to give 40 mg of the chitosan into your 200 ml of formulation. If it is your dose is 5 mg, so you have to calculate accordingly. So your syrup is you are adding as a vehicle, okay? So there is no problem until unless your green synthesized chitosan nanoparticles are safe, you can give as much. As per my requirement, so there is no cytotoxicity comes with the chitosan. But you after synthesize your material, your chitosan should not show any toxicity. Okay, so it will not depend on the chitosan. It will depend on the drug loading into the system. Okay, so if you have any further question, you can ask. We are happy to answer your questions. Yeah, any other questions, Rushikesh?
No, sir. There are no other
No.
questions.
Yeah, okay, fine. We have few questions from the uh, Google form. So we consider few more questions. So one person asked the question, but that is not question is not proper. So he's asking about novel drug delivery systems. So yeah, I think specific. We request you to ask a specific question so that we can come up with the specific answers. Okay. Yeah. So yeah, another questions. Sir, please guide us about the career opportunities in novel drug delivery systems, and which course of post graduation should we prefer further? Yeah. So I request you can, if you want to work in the novel drug delivery system, see after your B pharmacy, you should must appear the through GPAT, or you can give even for the GATE exam also, right? So with GPAT, you can join in into the M pharmacy. If you have a GATE exam, you can join into any type of MTech courses, MTech pharmaceutical technology, and you can even, I suggest you prefer attempt the NIPER exam also. So that will also will helpful for appearing the further studies. Now, in coming to that, okay, so yes, in all the fields like M-Pharm, I request you if you want to pursue in the novel drug delivery system, pharmaceutics, okay, and there are the institutes which they offer the MTech pharmaceutical technology, right? Even there are the institutes are give the degree on MTech formulation technology also, right? So there are the pharmaceutical formulation technology courses. So like formulation is one of the preferred platform if you want to go higher studies in novel drug delivery system. Even though if you opt like MTech pharmaceutical biotechnology in any IITs or any other institute, still you can do some project work on novel drug delivery systems. So that will help for so pursuing the higher education. And yes, and if possible, try to work on some like the industry-based projects during your masters. Otherwise, which should be helpful for industry. So definitely you can get an experience in industry after taking a 1 year, 2 years of industrial experience. You can further move for the PhD also. That will also very good area. So my conclusion is that you can opt any pharmaceutical subject which will help for the pursuing or learning in the novel drug delivery systems. Okay, by through GPAT or by using of NIPER, even in our institute also we have in the entrance exam. So if you clear that, you will get an admission in the MTech, but we requirement of the GATE that will give the fellowship. Yeah. Any other question, Rushikesh? Any new questions?
No, sir. I cannot see any.
Yeah, fine. So I request participants, please ask few more questions on the subject. Like so far, we have completed 4 weeks. Okay. So if you have any doubts in the 4 weeks, you can put it in the chat box. Okay. So we have few more questions. I am synthesizing some heterocyclic molecules for potential biological activity, how it will be used for the drug development. So, but you should specifically answer this question. Yes, you are synthesizing molecule, but what is this activity, right? So that you need to mention, because of course we have so many molecules that you can synthesize, but what activity you are expecting? For example, you are making some molecule which is for the anti-cancer, okay? Then definitely you should go with the cytotoxicity studies. I prefer that. And if you are going for the any antiviral or anti-infective applications, so you need to prove the activity first, okay? So if the activity is good, okay, and if the efficacy is good by doing of in vitro studies, okay, so then you look for the some in vivo studies, and not only that, you need to compare with some standard molecules, okay, so that should be show equivalent efficacy, right? So therapeutically equivalent, okay. Once it is therapeutically equivalent, what you can do, you can convert your heterocycle molecule into some formulation which may be conventional, either it may be tablet or capsule or any type of formulation, and compare with the marketed product. Okay, right? So if you are still you are getting enough activity of your molecule, prove that biological activity of your molecule. Once it is done, then go for the product development. Okay, right? So you can take that, synthesize the molecule, and screen for the biological activity. Once it is done, compared with the standard active molecule. For example, if you want to show the anti-cancer, take some related or relevant anti-cancer molecule and see the activity. If it is coming very good in the level, then go for making the formulation, right? That is again comes into the product development. Once you're done with in vitro studies, prepare a formulation and perform again in vitro studies with the formulation. And further, you can do ex vivo studies, and then you can go for the in vivo animal studies, which is called as preclinical trials. You can done with various animals, and you can initially see the pharmacokinetic studies, then go for the pharmacodynamic studies. If you're showing very good results, if your data is showing positive results, then you can consult into some the industries, okay. They will also, if they are interested, they will come and they will invest the money for the human clinical trials. So that is very big story, because we have various steps in the clinical trials. It go from phase 1, phase 2, and again phase 3. So it takes a lot of investment and it takes a lot of time. Okay. So this is a story of the product development. Okay. I hope I have answered your questions. Yeah. Anything, Rushikesh?
Sir, there is
Any
one
new?
question.
Yeah, please.
Sir, can I compare green synthesized chitosan nanoparticle syrup with zinc oxide nanoparticle syrup, or should I again microencapsulate zinc oxide nanoparticles with polymer?
Now, what is question? First, he prepared a green
Synthesized
synthesized.
chitosan
Uh-huh.
nanoparticles.
Yeah. Then
Then they
compared
have
with,
converted into a syrup.
okay, that already we have answered, right?
Yes. And
So what
further
is
he is
the zinc
asking
oxide?
with zinc oxide nanoparticle syrup, means they
Uh-huh.
want to compare, I think.
Acha,
How we
both.
can compare?
Okay.
Yes.
Comparative in terms of, yes, yes, you don't no need to reveal your active molecule. What you can see, the ultimately the particle size, but both are, according to me, it comes in different categories, because you're talking about chitosan, that is a polymer. Again, zinc oxide is an inorganic material. Okay. And you need to think about what is your application. That is also ultimately very, very important. Okay. Now, when you are taking a polymer, it or maybe inorganic, and if you are looking for only, I don't know what is your application. It is you are looking for antimicrobial studies, then if you don't have any active ingredient into this, okay, well and good, no problem. But if you have an active ingredient, okay, which is a you want to encapsulate in any type of the system, either green synthesized chitosan or in the zinc oxide, okay, you need to see in which system which is showing high encapsulation efficiency, okay. So based on that, you need to go further. So it again depends on your active molecule. That is the first question. Okay, that is the first answer. Okay, based on the high encapsulation or high drug loading, you can prefer any system. Okay, now the question comes in another way, like the toxicity of the material, how much concentration I need to use. That is also very, very important. Like chitosan, as per my knowledge, you can go in high concentration, whereas zinc oxide, not like that. Okay, so it has a limitation, because whenever you use inorganic nanoparticles, it will induces the it will induce the toxicity, or you can say the redox system, ROS system in the body. Okay, so first you need to consider the total encapsulation in both system and then compare. Okay, and second thing, toxicity of your blank material, or you can say zinc oxide and chitosan. Compare the toxicity concentration. So whichever is high, that I prefer, even if I develop any formulation. Okay, right? So yeah, I can compare greens inside chitosan nanoparticle syrup with zinc oxide syrup. Should I again micro encapsulate zinc oxide nanoparticle? You can, if you are facing any toxicity, you can go with the micro encapsulation. Okay, but for comparison, you need to think about encapsulation as well as the safety of both materials. Okay, and further activity also. Yeah. You can do microencapsulation. That is my answer. If you have problem in any with the zinc oxide, you can do the microencapsulation by using various polymers. Any question, Rusikesh?
No, sir, that's all.
Yeah, can we get some questions from that our what is that uh,
Forum.
the from the forum?
We have already responded to all the questions, sir.
Yeah, still we can discuss if there are the critical issues or minor things we can discuss in the forum. Anything you pick up till people are getting. I hope there is no questions. Yeah, we request you, please respond in the chat box and suggest your questions or whatever doubts you are facing. Even any difficulty in the lectures also, in understanding even the quality of the presentation. You can give some feedback also. If everything is going well, we'll continue with the same pace. Rishikesh, can you find some questions? Whatever comes in your mind. So far, we are started with the first week is introduction to novel drug delivery systems. So in the first week, we discussed on the fundamentals of drug delivery systems. So just I am elaborating. If you find any difficulties in these sessions, we can discuss. We are happy to discuss. And we discussed in the first week fundamentals of drug delivery systems. And also we discussed what are the polymers used in drug delivery system and the selection of polymers. And then we discussed on commonly used polymers in drug delivery systems. So if you have any doubts in the assignment also, just you can go through your NPTEL login portal, and then you can put and copy paste if you have any difficulties in the answering or any problem in the assignments. The second week, we have discussed nanoparticle as a drug delivery systems. So there we divide into 2 weeks, that is week 2 and week 3. In week 2, we discussed on the novel drug delivery systems, that is as a nanoparticle as a novel drug delivery systems. In second week, we discussed on the polymer-based nanocarriers, and also we discussed on the lipid-based nanocarriers. It's a very extensive topic. So then we split it into again week 3. So there we discussed on nanostructured lipid carriers. Also we discussed on vesicular drug delivery systems. We discussed on ethosomes and also transferosomes, bilosomes, all types of zones we have discussed. And later we discussed on the evaluation of the nanocarriers and then inorganic nanocarriers. Yeah, so week 4, we discussed on the targeted drug delivery system. There we discussed on the basics of the target delivery systems. So what are the various approaches of target drug delivery? We have discussed on the types of target drug delivery system. We have discussed on the tumor targeting, brain targeted delivery systems. So yeah, so if you have any doubts, any questions on these 4 weeks, kindly let us know. We are happy to help you. Yeah,
Sir, I have
Rishikesh?
some questions here.
Yeah. So someone is responded. Wait a minute. If I have to present a research paper related to nanoparticle targeted drug discovery, what are the common questions expected from the audience? Oh no, should not come from the audience. Yeah, because you are working on research paper, right? You should design your hypothesis first. Okay. So related to nanoparticle and target drug discovery, what are the common question expected? In research paper, is story is different. When you are writing a review paper, okay, so such cases, you can think about the audience like a readers, okay. So you can understand what is the gap between existing literature and from your literature. Based on that, you can elaborate. That will help for the readers. Whereas research paper is completely different. It is not depend on the readers. Okay, so it always depends on the your work. Okay, you should come up with some novelty. When again I am talking about research paper, you should focus on the novelty, right? So that is very important, right? So then again, if you know the novelty, right, you need to design your work based on the your current existing literature, or you can design a hypothesis. For example, consider in this way: I want to develop the siRNA, or you can say the DNA delivery, or you can say nucleic acid delivery, protein delivery. I know that these biological molecules are negative in charge. Okay. Now I want to complex with some positive charge to stabilize this negative charge and my positive polymer or some lipid. Okay. So I will use some lipid or a positive novel lipid, okay, which will improve the efficacy or stability of my nucleic acid. So what I will do the complexation, right? So this is a hypothesis, which what I'm suggesting in my hypothesis that, so when I use this positive charge, okay, this my negative molecule, it may be protein, peptide, nucleic acid, siRNA, DNA. When I complex it, okay, it will be very in the stabilized format, okay, which will not undergo degradation and would help for the it to release at right place, and this complexation will help from the all type of degradations. Okay, so that is our hypothesis. Now you from that you are expecting that, so once it is entered into the cell, it will follow the endocytosis mechanism, then it will release the drug, and then it will attack the or drug, nothing but biological molecule, then it will stop the synthesis of the DNA or whatever you can mention it. Okay, so that is the flow of your research paper, right? It not depends on the audience. Okay, so whatever outcome comes, you need to present in a with a good explanation of your outcomes. Okay, right? So completely different. So it is not depended on the audience. Okay, try to understand this. Whatever outcome comes, you need to work out with the different methods, and you take the data and present in a good format with the high resolution pictures and good writing skills and good English without having any like the grammatical spelling mistake. So that will help you. Yeah. Okay. And also one more personal suggestion is that you can put some 3 to 5 research papers of the same field. Try to imitate your work. Don't copy that. Just try to imitate your work and and take some the information from that research papers of the relevant work and try to imitate or perform your experiment. Later, you can work on your novelty part. Okay, so that is the best way of practicing. So imitating a published research paper, we can produce a novel or a good manuscript. Okay, yeah. So one more question. What is the scope of bone infection, site targeting nanoparticle, drawbacks, and future perspective? What is the scope of bone infection, site targeting? Definitely here, my recommendation is that, okay, so better to take some materials which I request to take some like bone grafting materials, like for example, the hydroxyapatite, okay, is one of the beautiful material which we can use in the bone grafting. Okay, so use that material. Even one more material, mesoporous silica. Okay, so these two material have high amount of drug loading. Okay, so because of this, surface area is very high, mesoporous silica. Okay, so you can use such nano carriers. There you can load your drug and you can target to the bone infection. Yes. And again, coming to the targeting, we have two methods. One is active targeting, second is a passive targeting. And what is active targeting? On the bone, you need to identify the pathophysiology and what are the receptors are overexpressing in the bone infection. Okay, based on that, take such ligands and label on your carriers. Then it definitely specifically go and bind to the your bone infected cells. Okay, right? So with that, you can do that. Other method is like taking a simple carriers, but the accumulation into different sites is one of the major issue that leads to the toxicity of your patients. Okay, that is one thing. And you are asking that drawbacks and future prospects. Future prospects already we have discussed. Okay, so that can be used like by using of advanced materials. Okay, by using a bone grafting materials, the residence time and accumulation can be increased. Now coming to the drawbacks. Okay, yeah. So see, when you are targeting for some organs, okay, like liver, okay, spleen, lymphatic, okay. In some other organs, the reachability or the permeability or retention at that particular organ is very high because your blood will reach into the that highly perfused organs like liver, spleen, okay, kidney. There you can reach, and even other organs also. The permeability or reaching into the bone target is one of the major limitation that I right now I have only this answer for the major drawback of the bone targeting. But if you come up with some active molecule, active targeting, definitely you can reach to the system. So one more question, dear sir, are you going to explain hydrogel based drug delivery system? Yeah, no, my answer is that because these are already, uh, uh, but some cases we discussed on the some notes we have covered, like not hydrogels, we have discussed on the gel-based systems. Maybe that will help. But if you're interested, kindly drop in. You can discuss in the forum. So we give you some article that will be helpful for your further studies. Okay. Now these hydrogel systems definitely we have very good application nowadays in case of ocular, even for the anti-cancer application, even with the various stimuli responsive hydrogels like temperature sensitive, right? Even the pH sensitive, right? So yeah, so for tumor targeting, these are one of the interesting formulations. But for this syllabus, we are not going to discuss on hydrogel. But in the stimuli responsive, we have discussed about detail on the hydrogels. Yeah. Any question, Rushikesh?
No, sir, I cannot see any further questions.
Okay, good. So yeah, can we have any other suggestions, questions from the participant, Rishikesh? Any common questions during the our forum? Any difficulties in there? Some of them ask some common question, right, regarding the MOOCs and advantage. Some they asked, no, some of them asked how much, what is the exam pattern?
Yes, sir. They particularly
Yeah.
asked for the exam patterns.
Uh-huh.
Yes.
So even the details very clearly mentioned in the portal, in the
Yes.
uh, program portal. There you can read. Still, you have what the dates are also given. The so enrollment already it's ended. Now exam registration is till 22nd August. So we request all the participants try to register with before the deadline comes. Okay. So exam date is the 1st November 2025. Okay. So the date, the time will be released through NPTEL team. Okay. So yes, exam will be on the online, but you have to visit to the NPTEL examination center. Okay. So there you need to answer, I think, 100 questions, right? So that is the requirement. Anything else, Rushikesh?
No, sir, I cannot see any further questions.
Any problem? Some of them initially they ask a lot of questions on the MOOCs and what are that? Anything else to discuss? Any questions in YouTube?
No, sir, no questions,
Live?
sir.
None, no? Is there any aspect of novel regular system for wound repair or healing? Yes, why not? Yeah, you can use as the Suresh Reddy explained on the mention on the hydrogels. With that also you can make. Even nanofibers is one of the good approach for the wound repair, wound healing. Now here what you can do, you can use some polymer, okay, which is antibacterial, or you can say the wound healing application, because we use various types of the wound healing patches. Okay, so you can use a material of, for example, consider chitosan is a one of the mostly used polymer in the wound healing patches or the wound application. Okay, so you can make a nanofibers of chitosan. It itself work as a antimicrobial agent. Now you can load some silver nanoparticles into that. Silver nanoparticle loaded chitosan nanofibers. Now the activity will increase. You can add wound healing drug also into that. Even people recommend starch also one of the best material for wound healing. Okay. So such cases you can load wound healing agent and you can provide antimicrobial, because wherever wound is there, right, you will see the further infection during the treatment. Okay. To avoid that, you can provide the nanofiber based patches, which may you can use the antimicrobial polymers, as well as you can use antimicrobial nanoparticles like silver nanoparticles, which will show the antimicrobial property. With that, you can enhance the activity of the antimicrobial application. I think, yeah, I don't see any further questions. Also, time also, we are about to close the session. Yeah, anything else, Rushikesh?
I cannot see any further questions from you.
Fine. So, yes, we are very happy to help you through live interactions. Okay. So, if you are if you face any difficulties, we are happy to help you, and we are happy to help you. And also, not only that, we will help to send some of the required materials if you are facing in understanding any limitations in this course. Okay, so myself and Rushikesh both are available, right? So you can put the questions in the forum for further topics. So in our upcoming classes, we are going to discuss on various novel drug delivery systems and any difficulties in the assignments also. we are here to help you, right? And if you need, like one person mentioned that on the like animations, so we have noted that suggestion. We send that link. I think that is JOVE, right? So now I got it, JOVE journals. Okay, so they will give you all type of videos. Okay, you can refer that JOVE. We will definitely share that link also through the forum. So you can refer, you will get all type of scientific research and education videos, okay, for laboratory and other things. Okay, clear? Yeah. So I hope, yeah, we are able to end the time. So if you have any questions, quickly we can discuss. I don't see any questions. With the permission of NPTEL team, sir, can we stop the session? We have 2 more minutes, but we have answered all the queries. Yeah, thank you so much. Thank you, everyone. Thank you, Rishikesh, and also the IIT Bombay team, NPTEL IIT Bombay team. Thank you so much. So
Thank
please
you so
come up with
much.
a new. Thank you so much. Please come up with the questions. We will definitely share the Google form. You can put your all your questions. Yeah. Thank you so much.
Thank you, sir.
Thank you, sir.